enamorados de la biología estructural. Vale, pues en esta última charla os voy a hablar de los estudios que hemos estado llevando a cabo concernientes a un factor epigenético que tiene como función silenciar genes. Y este es el Polycomb Repressive Complex 2 o PRC2. PRC2. El complejo está formado por 4 subunidades, de las cuales os voy a hablar especialmente de la ECH2 y de la EED. Esta es la que lleva a cabo la función principal de este complejo, que es la trimetilación de la lisina 27 en la histona H3. Eso es lo que hace. Este complejo hace eso, no hace otra cosa. Entonces, el ECH2 has a set domain that is a methyltransferase domain that imparts this modification on H3. So you can say it's a writer of what is called the histone code of modifications. But very interestingly, and this is going to be very important through the whole talk, it has a subunit called EED that is a reader of that modification. And not only does it read it, it senses it, then it allosterically activates the methyltransferase activity of ECH2. Okay, so this is very important. The complex is essential to silence whole genomic regions during differentiation to establish cell identity. It's also, because of this role, is also typically misregulated during cancer, where cells obviously lose their identity. And PRC2 is a major anti-cancer target now. There's a number of compounds being considered for clinical trials. The complex is not thought to often exist as these 4 core subunits. It always has additional cofactors. And the ones that we have studied the most are the ones called JARET2 and ABP2. Okay. These 2 cofactors give rise to a flavor of PRC2 that is often called PRC2.2. But in any case, I'm just going to be telling you about that one, so it's going to be PRC2. There is a very interesting interplay between these protein cofactors and the histone modifications that pre-exist when the complex goes. And this is going to be the main theme of what I tell you about today. Okay. So the combination of the cofactors that are bound to the complex and the histone modification context of the nucleosome that the complex is trying to act upon will define whether the complex is activated for methyltransferase activity. If that is the case, it will spread the modification. Oh, I forgot to tell you something. Okay, so this link between the writing and reading has, once it was determined to be the case, was thought to be very important for the spreading of the modification. So this complex doesn't come, do a modification, and leave. It tends to come and then kind of spread the modification. And the idea is because the complex is more active when there is already that modification. So if the context next door is one in which there's a nucleosome with this, that is has this modification, it should be able to spread it. And this was a concept that we proved structurally, and I'll show you that. In any case, when the activation occurs, the modification is spread, and as a result, the chromatin now becomes impervious to activation. It cannot, the transcription factors cannot act upon it, polymerase cannot act upon it, that part of the region, the genome is silenced. On the contrary, if the combination of cofactors and histone modifications inactivates the complex, now the modification is not present and chromatin is in a state where transcription of the type that I was describing you before occurs. Okay. So we started working on this again a long time ago, way before the direct detectors were available. And at that point, PRC2, which is about 350 kilodaltons, was too small. So this was at a time when the things that were being studied were microtubules, ribosomes, viruses, Cryo-EM, things with symmetry and big size. So Claudio Ciferri, when he was a postdoc in my lab, now he's the head of structural biology at Genentech, so a super big cheese. He decided to express these components in insect cells, reconstitute the complex, and he started with the 4 core subunits, and he couldn't get anywhere. The complex was just too flexible, and he couldn't do anything. And in fact, he was not studying it by Cryo-EM, he was studied by negative stain. This is a technique where you cannot get to the same kind of resolution, but it gives you very high contrast, and you can study things that are very small, even in 2010, 2011. So this is what he did. And he did a very good job at it, and he got a structure at 20 angstrom, which is typically as good as you can get with negative stain. Okay. So at that resolution, it's just a shape, but he was super resourceful. So because he had an overexpression system, not what we had for the human transcription factors, TF2D and things like that, what he did was to place GFPs or MVPs at N or C terminus, or GFPs in the middle between domains for all the different subunits. So he generated a huge amount of different complexes. And then he looked by negative stain, and if he had an extra blob corresponding to a GFP between one domain and another of ECH2, he knew that that position corresponded to ECH2 between those two domains. And doing that, he was able to localize the position of the different domains. So he was able to paint and say, there are 2 WD40 domains in this complex, and he was able to tell which one was which, where was the set domain, where was this and that. So it was pretty cool. And one of the things that he found was that the top lobe of the complex is made up of what the biochemists had defined as the minimal functional unit that can both methyltransferase, do the methyltransferase activity, but also be allosterically activated by the recognition of the methylated histone peptide by the ET subunit. So this was very cool. And a few years later, crystallographers, first for a fungi and then for PRC2 human, expressed those elements, which were the whole of ECH2, the whole of EED, and just the C-terminal domain of SUS12, and they were able to get crystal structures. And these were very beautiful because they showed how the allosteric activation worked. This is very important. I'm going to explain it to you. You have to understand it because otherwise the rest is not going to make any sense. Okay. So in this case, of course, they work with a peptide. Okay. So they have a peptide of the histone that is modified. It has a trimethylation in the lysine 27. And what they saw is that the peptide binds to the WD40 of EED, which is always going to be shown in cyan. And when it does, there is a region that otherwise is unfolded and they don't see within the ECH2 is in yellow. It corresponds to this region that they call the stimulatory response motif. So this is a part of the sequence of ECH2 that if there's no peptide bound, is invisible, it's unfolded. And when it's peptide bound, it falls over the peptide in the form of an alpha helix. Okay? So activated, alpha helix. Not activated, nothing. Why does it activate? Because this helix, when it's in the form of a helix, interact with one of the helix in the set domain, the methyltransferase domain, and give rise to the right orientation of the elements in the active site for methyltransferase activity. Okay. So this is what we knew. We went ahead because we wanted to get the structure of the full complex. Okay, that was still too difficult for the crystallographers. The top and bottom lobe move a lot. So we have to use all the tricks that I showed you about focusing on one, focusing on the other. But at the end, Vignesh, who now has his own lab at the University of Colorado Boulder, was able to obtain several structures. To do it, he had to do two things. Because this complex is one of those that breaks apart during the sample preparation because it interacts with the air-water interface and it unravels. Once we got the structure, we understood why. Because the top and bottom lobe interact through the burial of a hydrophobic interface that must be quite dynamic, okay? So this hydrophobic interface is competed away by the air-water interface. And you will see in a second, the bottom lobe falls apart. In any case, to get this structure, what Vignet did was twofold. One is he used a carbon support. So normally, you just form this meniscus of water, and the molecules are exposed to those two air-water interfaces. One thing that you can do is you can put a continuous layer of carbon, very thin, so that it doesn't contribute to the background. And then if it's hydrophilic, the protein will like to attach to it in a way that is likely not very disruptive. And then, it has a certain off rate, but some of the complexes will be attached, they will be not bouncing around. And in addition to that, he cross-linking, very likely using BS3, okay? And doing these two things, he was able to get several structures. This structure was with the full complex that have the four core subunits, ABP2 and JARET2, okay? And it so happened, this is going to be very important, you will hear this in a number of occasions. JARET2 is also a substrate for ECH2. So JARET2 gets methylated. It gets trimethylated at a particular site, and then that peptide can bind to the complex. And indeed, when Vignet did the reconstruction, there was a peptide that corresponded to JARET2, which gets methylated in the insect cells that have all the cofactors for the reaction to take place. And when it comes, we have it bound to the allosteric site. So we have a methylated peptide, we have an SRM that is stable, and we have an active complex, okay? Now, when Vignet, knowing this, truncated the JARET2 so as to truncate it internally so that it didn't have that lysine that gets methylated, he gets a structure that looks similar, except that there's nothing bound to JARET2, there is no density for the SRM, and then the conformation of the set domain is not activated. Okay? I'm not going to show you because that's quite old. Okay. So at the same time, Simon Popsell, when he was again also a postdoc in the lab, now he has his own at the University of Cologne, wanted to do something different. He wanted to work in a context in which the activation really was occurring in the chromatin context. So for that, he generated, think at that point, no one had looked at PRC2 bound to anything but peptides. And he not only looked at it binding to a nucleosome, he decided to do something a lot more interesting. That was to generate a dinucleosome where one is methylated and will serve as a substrate, and the other one is already trimethylated, and it could, in principle, serve as an activator. And this was to test the idea that the complex can bind, and by interacting with this one, it will be in the perfect condition to methylate that one. And that will support the idea of the spread. Okay? So he did this without JARET2, so that the JARET2 is not, where the only activation has to come from the nucleosomes. Okay? No JARET2, but this methylated in the nucleosome next to it. So he tested this by negative stain. So this is how PRC2 looks like, negative stain. You may be able to recognize the top, the bottom lobe. And then when he added the nucleosome, he got these two-dimensional, these 2D classes. And we were so excited because here we go. This is the complex and it's bound to two nucleosomes. You've seen them in different orientations. One is like this, one is like that. But here it is. This wouldn't necessarily have to have been the case, okay? It could have been that binding to both simultaneously was not compatible to them. It did. So we were super excited. However, we could not move into Cryo-EM because if we cross-link, the complex fell off the chromatin. And again, we now know why, because it turns out that the way the complex interacts with the nucleosomes is through interaction of lysines with the phosphate backbone, and when you cross-link enough to stabilize the complex, you are titrating away the lysines that are binding to nucleosomes. Also, he couldn't use a carbon support because if he did, he only got this view. And we need more views, otherwise we cannot reconstruct. So he had to go ahead and do cryo-EM under conditions where we know the complex was not stable. So this is what he got. He got the top lobe, which is the one that interacts with the nucleosome, binding to the nucleosomes. But the bottom lobe was disassembled. Again, it's just because of this competition with the in the interaction. Okay? But he went ahead and he obtained a structure, because this is the top, is the part that is doing the right kind of the interaction that we were interested in. Okay? And the resolution was limited to about 8 angstrom. This is because this complex is super flexible. This is actually important because here you will say, well, it depends on what linker length you use. We switch and we use a range of linker length that are compatible with the typical type of length that exists between nucleosomes in the human genome, and between the fact that the symmetry of the nucleosomes and the flexibility of the lobes and the linker DNA, the complex could get engaged with all, which was really nice. In any case, this is to show you the interactions that we were expecting were there, okay? So this is the nucleosome that serves as a substrate. The histone is engaged, and we can see it going all the way into the active site. This is the nucleosome that is already methylated, and look, we see peptide binding to the allosteric site, and we see beautiful density for the SRM. So this is what we expected. This is a complex that is binding to both and is in the active site, just ready to methylate. So all of this interpretation, it was the resolution is about 7 angstrom, was done using structures of the nucleosome that we know, the tons of them, and structures of the PRC2 that we had both from crystallography and our own cryo. But interestingly, just one thing, because of course, sorry, I'm getting ahead of myself. This was the first time that the complex had been shown binding to nucleosomes, and we have 2 nucleosome binding sites. We had one that is the one engaging with the ED, the one that is going to provide the allosterically activating tail, and that interacted with the side of the ED through this, with the both gyres, through positively charged regions. If we looked at the other side, the side that is engaging with the Z domain, it with the ECH2, and that it was being used as a substrate, it involved a domain within ECH2 next to the Z domain that is called the CXC. And again, these were highly conserved, positively charged surfaces that were engaging with the nucleosome. But what I wanted to say is that, again, all of this interpretation was done by docking pre-existing structures in this, 7 angstrom density. And very interestingly, there was this density that is right here between the CXC, the DNA, and the tail of the histone that didn't have a corresponding structure. There was something there that looked, based on when we played with the threshold, very intense, like it could be an alpha helix. But there was nothing that could correspond to it. Like everything else, both the crystallographers and us have given a structure to the elements that were stable, that we could see. There were plenty of regions that were invisible. So these have to belong to some of them. So remember this. One thing that is very interesting is that the position of that density corresponds to a site in the histone tail that is where the lysine 36 in histone H3 would be. Why is this relevant? Why do you have to remember all these freaking lysines? Because there are a number of modifications that are inhibitory to the complex. And one of them is trimethylation of lysine 36. This is a modification that marks regions of active transcription, okay? And this is one that had been shown in vivo and in vitro to be inhibitory to the complex. So I'll get back to that. Because this was the next thing that we wanted to engage in. We want to be able to, the nucleosomes that we've used so far were unmodified, but we know that this complex is able to recognize, for better and for worse, to be activated or not, different modifications. And one that is very interesting is this. So PRC2 works in conjunction with another polygon complex that is called PRC1. And PRC1 has a ubiquitinase activity. And what it does is ubiquitinase a certain lysine in a different histone, histone H2. And this complex, it was proposed based on biochemistry, Jarrett2, is able to recognize that monoubiquitination. Okay? And is proposed that because of that, it is brought to sites where PRC2 has deposited this modification in the genome, and will then do the trimethylation of K27, which, by the way, certain forms of PRC1 also recognize and are able to bring to the site and contribute to the compaction of the chromatin. So it was very interesting. Now the next thing will be, let's look now into localization by seeing if we can see this complex interacting with the immunity. But of course, we didn't want to have to publish these structures where half of the complex is missing, although we put it there. So Simon and Vignesh joined forces to establish a methodology that had been developed by Bob Glaser and Bon-Young Han at the Lawrence Berkeley National Lab. And this is the idea. The idea is to generate a surface of high affinity for the complex. And I'll tell you what this can do. The way it's done is you form a lipid monolayer and an air-water interface, okay? And those lipids are going to be biotinylated. And in the solution, you put a streptavidin. The lipids are biotinylated, so the streptavidin will bind. And then you wait and you allow a streptavidin to form a two-dimensional crystal. I'll tell you in a second why do we care about forming a two-dimensional crystal. But in any case, you can put now this monolayer of a streptavidin on an EM grid, and you can use biotinylated samples to make them bind to the streptavidin. And then they will be bound to the streptavidin. You can concentrate them. If you do the biotinylation right, you will get random orientation. I can tell you more about it if you ask. And also, it will, in principle, keep things away from the air-water interface, which was what was the most important part. So in our case, the simplest thing was to take a nucleosome and to biotinylate it and wire. You can bind it to the grid with this streptavidin monolayer. You can then add the PRC2. You wait, let them interact, wash off anything that is not bound, and then blot and get your structure. And this really worked. So I didn't put the slide that show why you get a two-dimensional crystal. The reason why you get a two-dimensional crystal. Yeah, I'm there for the physicists in the audience. Let me see. Oh, no, I truly removed it. Okay, never mind. So the reason why you get a two-dimensional crystal is that when you take your images, you have your complex, but you have this background of all the streptavidin. And that's not good, okay? But because the streptavidin is crystalline, if you, in the computer, calculate the Fourier transform, you get something that is the Fourier transform of the streptavidin plus the Fourier transform of your protein. The Fourier transform of the streptavidin is concentrated on Bragg reflections. Reflections like when you look at that diffraction pattern. So you must then out. Then you Fourier transform back. This is like summation and multiplying and dividing. They're inverse things. It takes you back to an image where now you preserve all the signal for the PRC2, but the streptavidin is magically gone. This is really great. That's why you get into them. In any case, I'm going on and on about this because it works beautifully. So this is the structure that Vignesh obtained. And it is the highest resolution, is beautifully preserved. There's no cross-linking, there's nothing. And we see the top and the bottom is really lovely. Okay, so what you see here in orange is the streptavidin, and what you see in magenta and the other thing in red are the elements in Jarrett2 and in ABP2 that recognize the monoubiquitination. And some of these elements have been predicted based on biochemistry, but half of these sites and the involvement of ABP2 had not. So this now gave us a structure that was, really nice and had this information. And of course, now we have a structure with a nucleosome and a high resolution. So what can we look at? the thing that we didn't see before. And indeed, it is a helix. And we call this the bridge helix. You remember that there was this density that we saw for the dinucleosome, but it had never been seen? It's only present when the complex is bound to nucleosomes, because it falls on nucleosomes. It's a region, it's the second region that I tell you about, that in the absence of something important is unraveled, but if it binds to it, it falls into a helix. This is a helix that is very positively charged on one side, and it interacts with the DNA. We call it the bridge helix because it is between the set domain, the nucleosomal DNA, and the tail. So very critical to set up the right geometry that basically channels the tail all the way into the active site. And this tail is beautifully seen. We see every single amino acid. And the region that binds this tail all the way to the active site, including this bridge helix, is very highly mutated in cancers when there's mutations in PRC2. Okay? So this is the second element. Remember, unfolding, you can switch between folding and unfolding depending on what it's bound to. All right. So I just showed you that ABP2 with these two cofactors can recognize the ubiquitinated nucleosome, and this is a way of bringing it to the site. It doesn't increase the affinity very much, but it gives it a specificity in terms of retention at certain genomic regions. And then I also show you that this modification in trans, okay, when it's in the adjacent nucleosome, can activate the complex, okay? So this means, imagine the complex is brought to a certain site, it starts methylating, and then it has this spreading capability. It will methylate the whole genome. So something had to stop it. And I mentioned that to you before. What stops it is these marks. The trimethylation of K36 and the trimethylation of K4, when they are present, makes the complex not being able to act on that nucleus. This had been demonstrated both in cells, through genome analysis, and in vitro. Okay? So in the genome, this K4 trimethylation covers the region around the promoter, and this other modification, the K36, covers the body of the genome. And this is why PRC2 doesn't act on areas that are transcriptionally active. Okay, so this is like a summary. Okay, crosstalk between modifications and the cofactors. Okay, so I told you this is great to have ubiquitin in the substrate nucleosome is great. It acts in cis. It makes the complex go there. This modification acts in trans and allows the complex to be very active on the nucleosome next door. If you are GREC2, is an activator. I told you, because it can act as an allosteric activator. So it can work together synergistically with the ubiquitinated nucleosome. It has no additional effect if there's already chromatin around that is methylated, because it can already be activated by that means. Okay? And then there's these two modifications that act in cis. Okay? So the nucleosome that contains them cannot be trimethylated by PRC2. And we and others have shown that in the presence of GREC2, which activates the complex, this negative effect is reduced. So just keep this in mind. So I'm going to give you what was the landscape of what we wanted to do. There is a whole range where the complex can have basal activity, some activity, not very much, or it can be inhibited, or it can be allosterically activated. And we had some structures. We had the structure of the GREC2 bound to, of PRC2 with the methylated GREC2 that is allosterically activated, and this is even a little bit better if that nucleosome is ubiquitinated. Then we have this one where if you don't have the GREC2 and there's no adjacent nucleosome providing modification, you have this kind of basal activity. But there's all these other things. There is the inhibited nucleosomes with either the K36 or K4 trimethylation, and there is those nucleosomes, but in the presence of GREC2. So how does this work? What are the structures? And this is what Trinity cookies a graduate student in my lab did. And just to, what are the structures for these things? Just let me tell you that all of those nucleosomes are bound with very similar activity, with very similar affinity. And this is because, remember, the main binding energy comes from those positive charge interacting with the nucleosome. These other modifications in the tails are in the noise, okay? So it is something else. Okay. So Trinity started by looking at these nucleosomes, and we knew something. We had an idea of what would happen, and I'll show you. You remember I told you, even when we didn't know that the bridge helix was a helix, we knew that was sitting very close to the size of lysine 36. So this is just a blow up of that area in two different views. This is with the density, so that you believe that we have the resolution to see this. That lysine is in a pocket that brings together the tail, where it is in the protein and the nucleosomal DNA. So it says the right geometry for tail engagement, and it's actually interacting with itself with the phosphate backbone of its own nucleosome. Okay? There is no space there for 3 methyl groups. So something has to happen. And in fact, we could see in our analysis that PRC2 would bind into the nucleosome, but when we did any averaging, we just show a blur. And it's like, yeah, it's binding to the nucleosome, but it's not binding in a specific way. All right, so the idea that PRC2 is actually sliding on the nucleosomal DNA was confirmed with the next study, which was to look at PRC2 when there is a little bit of activity, because PRC2 because JAREC2 is activating the complex. So what happens in that case? What happens in that case is that we see two structures, Trinity sees two structures, okay? One that she called tail engage, and this is one in which the complex, you can see the density coming from H3 going all the way into the active site. Okay. And then there is this one in which there's no tail. The tail is not binding, okay? When it is bound and she can look at it, she can see that the tail has reorganized a little bit, and this lysine has moved to a different position, so it's no longer contributing to the binding to this arrangement with the DNA, but it's a region where now those methylated groups can be accommodated. Okay? But this is happening only in a part of the complex. It's like the complex is binding and letting go because it's not binding very stable. So there is an equilibrium that is happening there, so that the complex has less of a chance to actually do the final trimethylation, which is what we observe. Okay? Now, when Trinity superimposes the structures on the nucleosome, the two of them, it turns out that the PRC2 is positioned in two different places. So in blue is the tail engage, which is very similar to what Vignesh saw with the ubiquitinated nucleosome. And the gray one is the one where there's no tail engagement. And when she looks into the detail, this is what has happened. This is looking down so that we really see the bridge helix. This is the bridge helix in the case where there is a tail. And there are regions in here that are interacting with the tails, and regions in here that are interacting with the nucleosomal DNA. When the tail is not there, the helix moves several turns. It slides past. And the surface, some of the surface that was interacting with the tail is now interacting with the nucleosomal DNA. So it is a sliding pass. And this is something that she showed with this morph, so that you see the extent of that motion, okay? When the tail is not there, the complex can bind equally well to any place along the DNA just by shifting that bridge helix. Okay. So what we think is happening is that when you have JAREC2, which puts the complex in the right conformation to try to engage to the tail at the active site, we have this unstable tail engagement that when PRC2 is not there is complete. So there's no engagement. And the PRC2, is able to bind to nucleosome, but it doesn't bind in the right place, and it doesn't engage the tail. Okay, so that's how we explain the effect of this trimethylated K36 in the low activity of the complex. But what about K4? K4 is so N-terminal that no one has ever seen it because it's so very far from the active site where the K27 is seen, and then we see a few more, and then the tail is just flopping around. So how can this be doing anything negative to the complex? Okay. So she studied these both in the presence and the absence of JARAT2, where you have some medium or some very low activity. I'm going to show you first what she sees when JARAT2 is included. She sees a mixture of states. I'll tell you what the other one is in a second. But the predominant one is this. Is one state in which JARAT2 is methylated, is there, is present, it has bound to the allosteric site, we see the SRM, and everything is normal, okay? But there are other states, one of those states is the one that is prevalent when you don't have JARAT2. So when you don't have JARAT2, there's no JARAT2 bound, there is no SRM, but actually, we see extra density. There is extra density in the pandemic pocket, and there's enough resolution that we were able to see this is the K4 trimethylated. The K4 trimethylated, which is supposed to be an inhibitor, is binding to the allosteric site, but is not folding the SRM. So this is it, you know. In one case, the JARAT2 is able to bind with this trimethylated peptide, stabilize the SRM, give rise to a competent complex. In the other, there is a peptide that binds that is also trimethylated, but there's no SRM, right? So it is occupying the allosteric site, but it's not doing the activation. This is an allosteric antagonist. So how can it be? So this is this structure now in ribbon diagram where we just superimpose this peptide. And I hope you can see that this peptide has a much more extensive footprint that includes regions that interact with the helix. And this one, if anything, antagonizes. It has a clash, an steric clash. So this is a completely different mechanism. This is acting on that very interesting site on the complex that is normally used for allosteric activation, and this time occupied, but without activating. It's an allosteric antagonist. So that's one mode of interaction, while the other one was just not being able to engage. In one case, it engages, but in a basal activity kind of a state, and in the other one, is not able to easily engage. Okay, questions? Because I'm going to tell you a different story too.
Is the RC2 located in subthalamic region?
No. So PRC2, where the different cofactors are, has not been studied. This is one of the things that we want. So they typically look for the presence of ECH2 or ED. These complexes are present in chromatin regions that can be activated or not in the ones that have to be constantly replaced. So not telomeric regions, not red transposon regions. These are regions that will be activated or not depending on the cell type. Yeah.
Esta doble marca, digamos, que tienes en esa región con una posibilidad de tener complejo represor y al mismo tiempo la historia activada en los estados pluripotenciales se produce como un marcaje bivalente.
Con que, sí.
¿Cómo abres este marcaje?
¿Cómo pasas de una? Well, so one of the things that we say is that you can have the complex there in a region of K4 trimethylation that is not active, but just based on our structure and biochemical data, if you now have JARAT2, it is a more powerful binder to the allosteric site, and it starts competing in a way. So I think that could be part of the mechanism by, expression, recruitment of JARAT2, something like that. That could be involved. Sí, eso es una de las posibilidades que nosotros proponemos. Parte de este interplay entre qué cofactores tienes y cuál es la situación de la cromatina a la que te tienes que enfrentar. Hay algunos participantes que puedo contar con que van a tener preguntas. Dime.
Disculpe, doctor, hay algo que no me quedó realmente claro acerca de CR2. Usted nos comenta que para que pueda metilar otras histonas tiene que haber una que ya esté dimetilada en el paciente, pero es estrictamente necesario.
Ah, ajá. ¿Hay más preguntas? Porque si no paso a la parte siguiente. ¿Sí? No. Vale. Pues resulta que ECH2, recordad, es la proteína que tiene la actividad metiltransferasa en PRC2, puede automecilarse. Ajá. Y esto fue descubierto aproximadamente en el momento en que nosotros estábamos sacando la estructura con el mononucleosoma ubiquitinado, donde por fin habíamos visto esta región que llamamos el bridge helix. Recordad. Al mismo tiempo, estos papers estaban siendo mandados a las revistas. Entonces, dos investigadores muy importantes en este campo, Tom Check y Danny Reinberg, publicaron back-to-back 2 artículos donde hablaban de la autometilación de ECH2 y de cómo activa el complejo. Y resulta que analizaron dónde estaban estos sitios de autometilación y están en estas 3 lisinas que están en el bridge helix. Una de las cosas que ellos demostraron es que la autometilación es in cis. So, a complex methylates itself. It doesn't methylate another copy of PRC2. It's self-automethylation, basically automethylation in cis. This makes sense from where these things are. Remember, this is the helix, this is the end where the lysins are. When not bound to nucleosome, this is unraveled, and this is where the active site is. In fact, so it's right there, where it can be methylated and have many chances of getting to a trimethylation state by itself. So this is important to understand that this is an activation, this is an automethylation in cis, is that the bridge helix undergoes these transitions, and when it's just floating around, it can automethylate. Okay, so these authors propose that regulation could also be in cis, meaning that the complex that has the automethylation is more active. In fact, what they propose is that the methylation regulates an autoinhibition. Okay, if you have a peptide that is right there, very close to the active site, it will bind. Once it's methylated and becomes a product, it will be released, and now the histone can come and be methylated. But I don't know whether regulation is there. What regulation is there? I mean, there's no regulation. There's no change in activity of a gene based on this. It will be a global thing, in any case. Once we saw where it was, because they didn't know, because that region had never been seen, we thought maybe the methylation facilitates the folding of the helix, which will facilitate the binding to nucleosome, or and do that either because they favor the binding to the DNA or they're favoring the binding to the tail, because the helix is involved in both. Okay, so we tested that through MD simulations, because it's not trivial to get a complex that is not automethylated. And so this was done with Carissa Sambon-Matsu, who is at Los Alamos National Lab, where they have big computers and they do big simulations. So I'm going to just summarize what she obtained, and is that the helix really has a tendency to fall apart when it's not with a nucleosome, but it falls apart when it's with the nucleosome, and the effect of methylation is none. It's undetected. There is a little effect on engagement to the tail that they saw as how often the tail was engaged when the helix is methylated or unmethylated, but it was very small. So we decided that maybe there were other things that were happening, okay? So we're structural biologists, what would we do? Let's look at it, okay? So let's look at a condition where there's no JARET2, because there are plenty of complexes in the cell that don't have JARET2, where there's no methylation around, which is what you were asking, The only methylation that exists, the only methylated peptide that exists is the one on PRC2 itself, okay? So these are the conditions. So we make nucleosomes, modify, just give them to it, and we got a structures of the complex bound to a nucleosome, and we also got this. You may think, yeah, this is the complex bound to a nucleosome. No, this is two complexes bound to a nucleosome. Two of them, okay? So there is one that is engaging with the nucleosome the normal way, and there is another one that is interacting with the linker DNA and with the other PRC2. This was unique, okay? So we refer to the one that is binding the nucleosome, is binding the tail, it has the bridge helix perfectly folded as the proximal PRC2. And we refer to the other as the distal PRC2. Now, one thing that I have to tell you, this is really flexible. This is both transient, is in an equilibrium between unbound, nucleosomes that are by themselves, nucleosomes that have one PRC2, and nucleosomes that have these two PRC2s. So the PRC2s are coming and going. So it's a transient, and it's super flexible. Because the linkers DNAs are flexible, and the regions of interaction between distal PRC2 with both the DNA and the PRC2 are through flexible loops. So it was very difficult, and it was very difficult to push the resolution. Okay, so there's a lot of motion. And at the end, again, this was happening after we had been struggling for this for a number of years. There was a new software within, a new function within a software that we have used before that was published called 3D Flex Analysis. And this splits the structure into bodies that move with respect to each other and then analyze it based on modes of movement. And you determine, I'm assuming that there's I don't know, 3, 4, 5 modes of movement. In any case, I'm just to give you an idea, it detected this kind of motion where this is the nucleosome and the proximal PRC2. This is the distal, and these are the DNAs. It's just wiggling around like this by a lot distance, by a big distance, okay? But having this analysis and breaking it into parts, we can analyze each one of the components, and we were able to get reasonable resolutions, very good for the nucleosome, pretty good for the proximal, and enough on the distal that we could look at secondary structure motifs, at folded or unfolded helices, okay? So we went from that to something where we can now dock in all the elements and come out with a structure, okay? So, because this view is very confusing, it's very good to show that you have two PRC2s and a nucleosome. I'm going to do a little bit of gymnastics to bring you back to what you've been seeing before and making a little bit. So, this is turned 90 degrees so that now the nucleosome is buried in the screen, okay? And now I'm going to turn that in plane so that I put them upside, and now with this, I'm just going to open them. So they're like this, they're facing each other. I'm going to go like this. Okay? So this is the proximal PRC2. You should recognize this. It's the PRC2 engaging with the nucleosome like always. Okay? And this is the other. This PRC2 is not binding to the nucleosome. It's binding with the two linker DNAs. And what you see marked with a circle as regions of DNA interaction. This is the classical that uses the CXC domain and the bridge helix. This is one that is binding on the side of the ED and resemble what Simon saw when the complex is interacting with an adjacent nucleosome that is methylated. So we knew there was this positively charged surface there. And then there is a third one that no one had seen before that involves SUS12. Okay? It's the second site in which the distal PRC2 binds to the linker DNA. And then what is marked with the red asterisk, this is the region that is involved in protein-protein contacts with this region. So I've opened them, but these were touching each other. And this is the ECH2 in the distal PRC2 and the EED of the proxy. Okay? And then there is another contact that is between two regions of SUS12 that come together here and there. All right? So in our analysis, we get 3 PRC2s. We get the proximal PRC2 that is binding to the nucleosome, the distal PRC2, that is the second one, and then we have the complexes that is just one PRC2 bound to the nucleosome, which is here. Okay, so that is what we have in our data analysis. And I'm going to look at these 2 elements that can be either folded or unfolded. So one is the bridge helix. Okay, so the bridge helix here, there is very clear density, is low density, low resolution, but it's very clear density for the helix. In this case, the distal PRC2 is not interacting, in that element is not interacting with anything, and there's no density. It's completely empty, okay? And in this one, in the monomer, the complex is engaged with the nucleosome, the helix is perfectly happy, folding and binding to the nucleosome. And now I'm gonna look at the interesting one, which is the one that gives us whether the complex is activated or not, okay? The allosteric site. So in this complex, there is plenty of density for a peptide. There is beautiful density for the SRM. That is totally empty, either here or here. These 2 complexes have nothing activating them, but this one does. Okay, all right? So one is stimulated, the other ones are not. Okay, so going back to our dimer, there are 2 interactions. One happens between regions in cis-12s that have complementary charge. It involves loops. The other one is the one that is really interesting, because this is the one in which the region where the automethylation happens is present, but in that complex is not binding to a nucleosome, is not folded. Instead, it has reached and bound the allosteric site. And that's why the SRM is folded. So one PRC2 is activating in trans the other PRC2, forming a complex that only exists in chromatin when one of them is engaged with a substrate and with a substrate nucleus. Super cool. Okay. So we are proposing that in these 2 PRC2s, there's a separation of functions. So we wanted to test this biochemically. Okay. So this study was done in collaboration with Simon now at his lab in Germany and through his student Igor. We generated a number of mutants. So this is concentrate on what this is. Okay. So one is a mutant where we mutate this surface of interaction. So remember, there are 2 surfaces of interaction. One, we just call it dimerization, and we mutate it and it should weaken because we weaken one of the interactions. And the other one is the one that involves the methylating peptide. So in that case, what we did is to mutate the lysines to arginines. They cannot be methylated, but otherwise they're also positively charged. They should not be able to bind to an allosteric site. But for the rest, it will be totally fine. The complex should be able to bind nucleosomes and do activity at basal level and all this. Okay. And then we generated one more mutant. Okay, so those are 2 types of mutant. I'm going to cross them with this. So, by the way, so the mutant that has this, I'm just showing here, is a mutant that has some activity. The activity is shown here. Is an antibody, is a Western with an antibody for the trimethylated K27. And this is the complex by itself has this activity, what you see right here. Now, okay, now we generated another mutant. This is a mutant with the region in the CXC that contributes massively to the binding to the DNA is mutated. They had been proven before by a collaborator of ours that it cannot bind nucleosome and it has no activity on nucleosomes, okay? So this is it. This is the mutant. It's a mutant PRC2 that for everything else is fine, but it cannot bind nucleosome through the normal site. And it has no activity. Now, we put the 2 together. This one, which is non-methylated, and it cannot act as an allosteric activator. And this one that cannot bind to nucleosomes, but in principle, it could do the allosteric activation. And we get this. We get an increase in activity of 50%. Okay? Now, we are saying that this is based in dimerization, and methylation. So we can now generate two mutants. This one, so in the one that cannot bind to nucleosome, in addition, we either make it that it's not automethylated or that it cannot dimerize. And when we add those, nothing happens, okay? There's no activation of the other complex. So I think this proves that there is this separation of function where one complex that can engage with nucleosomes can be activated by another one that dimerizes through it, with it, and brings in a methylated. So this is the summary. I'm ending here. So we think that in addition to any possible in-cis activity, there is a trans activation in which the complex is using the ED and the normal allosteric mechanism that I now described to you many times, okay? And that in the context of chromatin that does not have local methylation, the complex can bind, can engage with the nucleosome with the tails. It's okay. But it just has basal activity. But another one that is automethylated can now engage with it by binding to the local region, the linker DNA, and give them that methylation, okay? So that one complex is activated while the other one acts as an allosteric effect. Why is this significant? First of all, how many complexes do you know that can read and write chromatin and in the process become activated, and read and write itself and in the process become activated? Very cool. This activation via by modification in a trans way should remind people of what some kinases do. Okay, so receptor tyrosine kinases act this way. They methylate. They phosphorylate and then they become active by dimerizing. But in any case, it had not been seen or described for epigenetic factors. So what is the biological significance? We think that this could be the mechanism by which PRC2 is activated when there's it's a way of initiation under conditions where there's no JARET2 because it's not expressed or what, okay? Now, what is the regulation there? The regulation comes because for formation of a dimer, you need high concentrations. Much higher concentration than just for a monomer to come and act on nucleosomes, okay? So there are ways in which JARET2 can be concentrated. It has been proposed that maybe through interacting with RNA. It has been proposed that through inclusion into phase-separated domains and things like that. So local, something that brings PRC2 without activating it, but it brings it to the site, increasing the local concentration, will allow this form of starting the methylation process. Now, once this has happened for a while, the other mechanism will take over because, of course, it requires now 100% of the PRC2s can act as a methyltransferases rather than half of them having to be sacrificed as allosteric effectors. So once enough density of this modification exists, this mechanism takes. Okay, so this is the final story, very mechanistic today, this second half. I mentioned most of the people. So for the earlier studies that are published, not in bioarchives, I told you about Claudio, Simon, and Vignesh, and Vignesh was actually helped a lot with the streptavidin monolayer and getting the structure of the ubiquitinated nucleosomes by two Berkeley undergraduates that are now doing the PhD at MIT and Hopkins. Trinity has been working on this inhibitory post-translational mark together with Ali, who is a Berkeley undergraduate right now. And then the work on the dimer was initiated by Simon, and it was continued by Paul, who now is working for a biotech in the Bay Area. And the biochemical assay that I showed you was actually done by Igor in Simon's. And I haven't told you, but all our studies are funded by NIH and HHMI. And in the case of microtubules, we have new projects that I haven't told you about that are funded by the European Research Council. Be aware that the European Research Council has this grants called synergy grants that involve groups of people, typically 3 or 4, and that for the last 2 years, they changed the rules so that one person of the group may not be member of the European Union. So if you have collaborators in Europe and you can think of a project that is highly synergistic in nature, where you do different things that complement each other that could have a high impact, this is big money, like big. So, and with that, thank you very much for your time.
3 preguntas.
Number 1.
Número 1.
Número 1.
No existe ninguna diferencia cultural entre la PRC2 distal y proximal. Son exactamente iguales y
Bioquímicamente son exactamente iguales, lo único que cambia es el contexto. Entonces, conformacionalmente son muy diferentes. Una tiene las 2 hélices plegadas y la otra tiene las 2 hélices unfolded. So, están en el mismo estado bioquímico y tienen estructuras tan diferentes como hemos podido ver en PRC, porque uno es activated y el otro no.
Y entonces la activación es la que determina el papel que van a jugar.
Exacto. Sí.
La otra pregunta va relacionada al ciclo de metilación.
Sí.
¿Tiene algún papel en la metilación? Se ha visto que algunos ciclos, cuando son ubiquitinados promueven las modificaciones transcripcionales de otros sitios,
Exacto.
como puede ser la metilación. ¿Tiene aquí un papel o no?
Exacto, sí. Esa ubiquitinación, que es en un lugar muy preciso, porque hay otras ubiquitinaciones que están relacionadas con activation, o sea, esta en particular, que es la que establece PRC1, tiene como función traer al PRC2 para que deposite la trimetilación de la K27. O sea, que es algo que permite localizar la segunda función en sitios determinados. Hay un link entre las dos. Ya.
Y la tercera está relacionada con polimorfismos. Se ha visto que varias metiltransferasas presentan polimorfismos y estos están asociados sobre todo a cáncer. En particular, con esta metiltransferasa, han buscado si estos sitios se encuentran mutados en enfermedades como cáncer. Sí,
esto es el tipo de cosas que estamos haciendo ahora. Tenemos 2 colaboraciones, una con un grupo en Barcelona que ha descubierto un link en un tipo de cáncer que tiene que ver con un splicing de SUS12 que cambia la cantidad de PRC2.2, que es el que yo os he dicho, con el PRC2.1, que utiliza otros cofactores que no son JARAT2 ni ABP2. Y estos los estamos estudiando ahora y estamos estudiando las distintas formas, con el splicing y con el non-splicing. Es muy interesante. Pero también ahora uno de los factores del PRC2.1, que os estoy diciendo, otro cofactor que hemos ¿se está grabado? Ostras, ok, bueno. Se ve que su overexpression se correlaciona con ciertos cánceres y hay una serie de mutaciones en regiones que son muy interesantes desde el punto de vista de lo que vemos nosotros, que también tienen una relación. Así que no, eso es el tipo de cosas que ahora estamos viendo.
Gracias.
You mentioned that in your model there is DNA linker.
Yeah.
What about the histone linker? Which one?
Yeah, very good. Okay. So one of the things that we thought when we were trying to get more complexes that have the dimers to stabilize the dimer was, what if we put H1 and H1 stabilize the linkers, and this favors the binding of the second PRC2. So we've studied PRC2 with nucleosome and with H1. Okay, so this is the structure. This is H1. It sits there. It fixes. Es un poco pequeño, pero bueno. it fixes the position. So this one is the H1, okay? It fixes the position of the direction of the linkers as they come out. Okay. So when and by the way, we can get it even at higher resolution if we just look at the nucleosome with H1. But in any case, we have those structures. Cuando ponemos H1 en condiciones donde normalmente tenemos el dímero, tenemos dos cosas. O H1, como este, con un monómero, o el dímero sin H1. O sea, nunca vemos que haya H1 cuando formamos dímero. Nunca. Y eso tiene que ver con el hecho de que en presencia del H1, el ángulo al que salen los linkers es incompatible con el ángulo que necesitamos para formar el complejo. Entonces, esto nos indica que en una región que es alta en H1, esta forma de activación es very unlikely.
Doctora, me llamó mucho la atención que muestra cómo el se une o interacciona con la cromatina. ¿Qué tan estable es esa interacción? ¿Puedo platicar un poquito más de eso?
Nosotros sabemos que PRC2 tiene varios sitios de interacción con el ADN. Son sitios que son tienen mucha carga positiva. Y lo que nos está demostrando este estudio es que además de poder unirse ADN que está curvado en el contexto de un nucleosoma, también se pueden unir con probablemente mucha menos afinidad a regiones que son lineales. Entonces, nosotros creemos que esta interacción existe porque cada una de esas cuatro interacciones, el distal interacciona con un linker, con el otro y con el otro PRC2 a través de dos conexiones, que cada una de esas es débil. Y que son las cuatro las que hacen, es una avidity effect. Entonces, por eso es por lo que el contexto es súper importante. Entonces, cada una de esas, sí están utilizando sitios que se ve que están positivamente. Es la geometría y el proceso es el avidity.
¿No han explorado el DNA metilado?
Eso también es una cosa que estamos considerando. Pero sobre todo en el contexto de otros cofactores que se saben que tienen especificidad o no, por metilado o no. Sí.
Entonces, ya tenemos estas tres metilaciones que nos dieron el paper de este particular. Ustedes han regresado a ver las estructuras que
Sí.
ya habían visto y si la resolución para ver algún estado de transición entre
Ya.
1, 2 o 3.
Sí. No, lo que hemos hecho es analizar. Es muy difícil hacer más spec y saber cuál es la methionine que está metilada o no. Entonces, lo que nosotros hemos hecho es utilizar, es un poquito indirecto, pero ayuda, ¿no? Utilizar péptidos, generar péptidos donde hacemos el trimethylation que nosotros queremos. Lo ponemos en una, en la otra, en la otra. Y luego añadimos péptido en cantidades más altas de lo que tenemos en distal PRC2, porque no tienes esta avidity effect, pero si lo añades, vemos que hay uno en particular, que es el 510, una cosa así, que es el más activo. Entonces, yo creo que los tres sitios están metilados en determinada cantidad, pero que hay uno que es el mejor para activación. Es indirecto, no lo podemos poner con nuestras estructuras porque es que tiene muy poca resolución. Y ese loop es flexible y puede que incluso tenga varias posiciones en las que le podamos ver. Entonces, es indirecto, pero por lo menos nos dice que una de ellas es más posible que sea correcta.
Ya sé que pregunto mucho, pero...
No, es que tú eres como yo. Yo en los seminarios estoy muy... Mucho galán. No, hago muchas preguntas, o sea que no te preocupes.
Me interesa mucho la autometilación de en el sentido de, bueno, si hay alguna forma de saber el ratio entre la cantidad de proteína metilada y la metilada, o si hay alguna
Nosotros
forma de
no hemos conseguido purificar PRC2 que no esté metilado,
que no esté.
pero sabemos que no está metilado máximamente porque podemos coger el PRC2 y añadirle los cofactores que necesita el centro activo para la metilación y vemos que la metilación incrementa. O sea que ya viene purificado, metilado hasta determinado grado, pero podemos forzarle aún más a que se autometile más.
Yo tengo una pregunta media loca. ¿Qué pasa en el espermatozoide donde no hay histona y están
Qué
las
loca, ¿eh?
protaminas? Y están las protaminas en lugar de las histonas.
Ni idea.
Porque
Ni
no
idea.
debería de pegarse esto.
Me parece
probablemente.
súper fascinante. Me parece una de las cosas más fascinantes que me han preguntado últimamente sobre el sexo masculino. No tengo ni idea.
A mí me gustaría preguntarle, en este contexto de la autometilación, por ejemplo, se puede conservar o se mantiene en condiciones, por ejemplo, de envejecimiento o en un estado más joven, por ejemplo, de la célula. ¿Es el mismo patrón que se podría encontrar o es diferente?
No se ha investigado. No se ha investigado el nivel que existe. Es una pregunta. Si pudiera contribuir de alguna manera al aging o una cosa así, o estás pensando. No sé. Estos procesos, un proceso como el de metilación es sensible al estado metabólico, porque lo mismo, utilizan estos cofactores químicos que si bajan van a reducir esta autometilación. Ni siquiera sé decirte cuál es el turnover del PRC2. Y tampoco sé decirte, porque no lo sabemos, si hay dimetilasas. Esa es la única manera en la que el regulation in cis could be regulated, ¿no? Que el estado de autometilación se cambie a través de estados metabólicos o mucho más inmediato de la presencia de dimetilases. Pero es que no se sabe. Para esta no hay mucha información de cómo se hace. You had enough of me? Okay. Un maratón. Pues me he quedado sin contaros lo del el Line 1 retrotransposon. Pero bueno, así tengo que volver a traer.
Por favor.